Thank you very much. It's a great pleasure to introduce to you today Dr. Sheila Peters from Merck KGaA, Darmstadt in Germany. She's currently the head of translational quantitative pharmacology there at Merck, and has a wealth of experience in the area of PBPK modeling. For more than 20 years, she's been working in this area, starting off early in her career at Cyprotex in the UK, moving on to AstraZeneca, and having developed her own models using the MATLAB platform. She's participated in a number of IQ initiatives concerning PBPK modeling. This has resulted in some excellent publications, and she has authored a book on PBPK modeling. And I was very excited to see in her bio that she's actually part of the FPIA ICH M12 group, which is a group focused on harmonization of drug-drug interaction guidelines. And I know that we're all eagerly waiting the outcome of that group, and it's good to see that we're in good hands with scientists such as Sheila. So without further ado, Sheila, over to you.
Okay, thank you very much, Steve, for that very nice introduction. Can you see my slides, all of you? I cannot change my, I can't, okay. So good morning and good afternoon, everybody. And today I'm going to speak on behalf of the Organ Impairment PBPK IQ Working Group on our work with the evaluation of PBPK models for organ impaired populations. The organ impaired PBPK working group members are drawn from 19 member companies under the Translational and ADME Sciences Leadership Group and the CPLG leadership groups. How do I can't seem to get rid of this anyway. So these 19 members are shown here, and the current affiliations of these people, some of them have changed because we have worked in this for quite a long time, for over 5 years, and in the meantime people have changed and their new affiliations are shown in brackets here. Just so if people have not already seen this paper, we have published the work that I will talk about today last year in CPT as a white paper. Before I go to the objectives of my talk, I would like to briefly touch upon the pathophysiological changes in renal impairment populations as well as in hepatic impairment. So here to start with the renal impairment is characterized by a reduced glomerular filtration rate as well as changes in the kidney size and gastric emptying time. It is also characterized by accumulation of uremic toxins, and this accumulation of the uremic toxins can in turn lead to reduced CYP activity, either by downregulation or by the direct inhibition of uremic toxins of CYP enzymes. Uremic toxins can also inhibit some transporters like OAT. It can increase the unbound fraction of the drugs by competitively binding to plasma protein or by inducing hypoalbuminemia in some patients. Thus, renal impairment by, through this alterations in CYP activity, transporters, as well as plasma protein binding, can alter the exposure of a drug that is not primarily eliminated by the kidney. So also those eliminated by the liver. So we use SimCip version 15 for our evaluation, but other platforms could have been used as well. It was just that all of us did not have access to the to other platforms, though all of us had access to the SimCip version 15. So that is why we have used this. But there are examples of this in the literature. For example, I have shown here one example where GastroPlus was used in renal impaired populations, obese populations. So SimCip version 15 captures the physiological changes in glomerular filtration rate kidney size, gastric emptying time. It also has a way by which we can take into account protein binding differences in this population. However, there are features that it does not consider today, and that is a mechanistic consideration of the inhibition of enzymes and transporters by uremic toxins. So this is taken care of in a non-mechanistic way, as by adjusting the CYP abundance rather than by actual affinity of uremic toxins towards these enzymes and transporters. So it is not mechanistically taken care of. Now, moving on to the pathophysiological changes in hepatic impairment population. Hepatic impairment in liver cirrhosis is characterized by a progressive decline in hematocrit, hepatic blood flow, alpha-1 glycoprotein, albumin, and also a decline in functional hepatocytes and biliary excretion. It is also characterized by alterations in the expression of activity of hepatic transporters and metabolizing enzymes, as well as duodenal CYP3A. So it's not just hepatic, it's also having an impact on the duodenal CYP3A. Liver cirrhosis is also characterized by the insertion of portocaval shunt, and this is illustrated by the figure shown here. Really want this gone? Yeah, okay. So this figure here shows a comparison of normal liver and cirrhotic liver. And what you can see here is portocaval shunt. And why it is coming? Because in cirrhotic liver, there is a formation of connective tissues and nodules, and that can lead to the obstruction of the venous flow. And the resulting intrahepatic pressure triggers the insertion of the portocaval shunt and releases this intrahepatic pressure. What does it mean for PK? This means that there is no hepatic first pass extraction for these drugs. So they can simply bypass the hepatic first pass extraction. And so you expect the exposure of the drug in systemic circulation to be quite high. However, this portocaval shunt effect changes with disease progression. So this is a compensatory mechanism, and it becomes quite inadequate with disease progression. And eventually what happens is that there is a effective arterial blood flow volume decline because of the obstruction in the liver. And because of this, there is a diminished renal blood flow, which in turn triggers the renin-angiotensin-aldosterone system. And the resulting hyperdynamic circulation ultimately leads to a reduced glomerular filtration rate and therefore to acute renal failure. And thus, you can see that liver disease can impact kidney function through this mechanism and alter the drug exposure for a drug having significant renal elimination. So PBPK models, in particular SimCip version 15, captures the pathophysiological changes in renal and hepatic blood flows, GFR, CYP abundance, albumin, alpha-1 glycoprotein, and hematocrit and liver volume. What it does not consider, however, is the potential altered scalars. Because what we consider is, okay, that there might be a change in liver volume, a reduction in the liver volume with disease progression, but every gram of this liver could also have a much reduced amount of the protein which is necessary for the metabolism, and that is not really considered. So that is assumed that we are already taking care of that somehow with a reduction in the liver volume. Don't seem to have, okay, there it is. Okay, so this slide shows the objectives of our work, but before that, I would like to talk a little bit about the regulatory recommendations for organ impaired populations and the motivation for our work. So organ impairment mediated drug accumulation can impact drug safety, and therefore regulatory agencies require a PK study conducted in organ impaired patients of varying disease severity for drugs that are primarily eliminated by the impaired organ and that are likely to be used by this population. So the disease severity is categorized based on measured estimated glomerular filtration rate or creatinine clearance for the renal impairment, and Child-Pugh classification, class A with a mild, class B with moderate, and class C with severe impairment, hepatic impairment. So these studies that are required by regulatory agencies, dedicated studies in these different classes of severity, they can be resource intensive. It can also take several years to recruit these patients with different levels of disease severity. And therefore, we think that there is a great opportunity for PBPK modeling of organ impairment populations to supplement the limited data that is derived from fewer patients, and with the potential to reduce the timelines and cost if models are validated for the purpose. However, validations of hepatic and renal impairment is not so much. It is very isolated data based on very few compounds, and these validation studies are not done systematically with a standardized workflow. And because of this, the regulatory confidence in PBPK models for impaired populations is quite low. The objective of our work was therefore to evaluate PBPK models for organ impaired populations using standardized workflow on a large data set with a range of clearance and elimination pathways. So with the results of this work, we hope to address two major questions. First of all, is the performance of the organ impaired PBPK models sufficient to rationalize the need, timing, and design of these organ impairment studies? And secondly, whether the performance is sufficient to supplement the limited data, clinical data, when recruitment is incomplete, as we had highlighted before, or is difficult, as in pediatric and oncology patients. So in order to address these questions, we considered a large data set from the 19 IQ Consortium member companies for 29 compounds with 56 hepatic impairment study arms. And in these study arms, there were 18 mild, 25 moderate, and 13 severely impaired subjects. And we also had 50 renal impairment study arms with 8 mild, 14 moderate, 25 severe, and 3 end-stage renal disease. So as you can see here, the distribution between the various disease severity subgroups are quite good, except with this end-stage renal disease where we only had 3 patients. And as you can imagine, this is not an easy subgroup to recruit. And therefore, these numbers are low, and which means the conclusions that we draw for this subgroup may not be valid. Okay, so I told you that we had used a standardized workflow, and that is what is shown in this slide. So what you can see here is the basic model being built using physicochemical properties of the drug and in vitro data for clearance, protein binding, etc. We use the clinical PK data from SAD studies, first in human studies, and also measures of pathway contributions, usually coming from mass balance data. And this means, what is the fraction metabolized, what is the contribution of the CYPs among this, and what is the fraction eliminated renally or via bile. Once the model is built initially, we verify and refine in an iterative manner using other PK studies that may be available, like multiple ascending dose studies or any other PK data that may be available. In addition, we also try to verify the model by using any DDI data that might be there already for this compound in healthy volunteers or in patients. In addition, if a compound is metabolized by enzymes with known genetic polymorphisms, we also did the simulations for subjects with different metabolic status, extensive metabolized, poor metabolizers, as part of verification. So once the model was verified, we then used it to predict the exposures in healthy controls. And if this exposure, predicted exposure, is within twofold of the observed, then we started to integrate the model with the OI specific, organ impairment specific parameters, such as blood flow, glomerular filtration rate, CYP abundance, that are specific to the population. And we also try to use the ex vivo protein binding data as input for the model. The in SimSip, we also have a way by which we can predict this protein binding, and actually we have tried to compare this predicted protein binding in organ impaired patients versus what was observed in the ex vivo, and found that the predictions were pretty good. But whenever the measured data was available, we always use the ex vivo, and we are confident that when this data is not there, SimSip would have done a good job in predicting that for the rest of the compounds for which we didn't have. Once we are happy with the model, then we try to simulate the organ impaired exposure impairment. I mean, the exposure in this population. So that was the workflow. All of the models were built in SimSip version 15, and they were validated by modelers from the contributing companies. The human ADME study data were available for most of the compounds, and that means nearly 75% of the compounds. I think I covered this. And the virtual trial simulations was done in the usual way, 10 trials with 10 subjects that matched the clinical study design with respect to dose, dosing frequency, age range, and gender ratio of the study cohorts. Prediction successes where a success was assessed using both the twofold prediction error and the guessed criterion. Now coming to the results. So this slide shows the AUC ratios of renal impaired subjects to matched healthy controls, and as well as the Cmax ratios of renal impaired subjects to matched healthy controls. So just to give you an idea, so the AUC ratio in hepatic impaired or renal impaired population is likely to be more if you imagine that the metabolic capacity of this impaired organ is much less, and therefore you have a higher exposure. So in general, you would expect that in the impaired population, organ impaired population, you would have a higher exposure, and therefore the AUC in the renal impaired or hepatic impaired to matched healthy controls would be greater than 1. But you can have cases where they are less than 1, because you can also have protein binding that is dictating the change in exposure in the organ impaired, and that goes in the opposite direction. And so what you see here is that the observed effects for renal impairment are rather modest. You can see here that the observed here, the range is pretty narrow, between 0.6 to 2.2. The PBPK predictions were generally within twofold, and this was for greater than 90% of the studies. So that is quite good. Especially when it is just a twofold. For the guessed criteria, yes, then there are a few more going out, but in general, the predictions are looking good for renal impairment. The accuracy of the predictions was very good for those compounds with the renal clearance greater than 25%. So when the renal clearance is substantial, significantly contributed by renal clearance, then those compounds are all here. None of them are outside the twofold. So that is quite encouraging, because it is for this type of drugs where the renal clearance is exceeding 20. I think now it's 30% in the recent draft guidance. It is like 30%, I think. So whenever the renal clearance is high, then the model predictions look good. So none of them are here outside the twofold. What you can also see is that there is a tendency to over-predict rather than under-predict. So for the twofold, you don't have anything under-predicted, but you have these couple of compounds. For the guess criteria, you have more of them over-predicted, but only one of them is under-predicted for the AUC ratios. For the Cmax, you find similar trends. There's nothing, the picture looks very similar between these two. I also would like to point out one the observed ratios, as we had seen, was quite modest, but then the exception was this compound. This is actually a metabolite for one of the compounds that is here. So of course, for a metabolite, you would expect the renal contribution to be much higher, and possibly because of that, you also have a high impact of impairment, and therefore quite high values of AUC ratio. So now to the results for the liver impaired subjects. So this shows the AUC ratios of liver impaired subjects to matched healthy controls. The closed symbols here in all of these three plots, they are all drugs with low IV clearance, and that is defined by this, that is the CLIV is less than 20 liters per hour. And these three plots are for CPA, that's the childhood class A, class B, and class C. That's the mild, moderate, and the severe impaired populations. So again, what you can see here is when the clearance is low, less than 20 liters per hour, these drugs tend to have lower AUC ratio. So you can see the shift of all the closed symbols to the left relative to the open symbols in all of these three pictures. So the closed symbols are to the left compared to the open symbols in all of this. So in general, they ranged from the AUC ratios ranged from 0.1 to 10. And the PBPK predictions were within twofold of the observed for greater than 70% of the study. So this is a little less. For the renal impairment, we had seen greater than 90% were within twofold. For the hepatic impairment, it's slightly less. So it's only within twofold for 70% of the studies. One notable thing is, you don't see any under predictions. Everything is, whatever mispredictions, they're all over predicted. None of them are under predicted, whether it is for CPA, CPB, or for CPC. There are no under predictions, and all of them are over predictions. And this tendency to over predict is more for the low clearance compounds. And that means you can see that it's only the closed symbols that are outside of the twofold dotted lines, whether it is the CPA, CPB, or the CPC. So it's only the closed symbols. And what you also see is that the mispredictions is also for much higher when you consider the level of severity. So here you have only one, and this is in mild, and with moderate you have more number of compounds, more proportion of compounds that are outside the twofold range. It's nearly 50% if you can consider the guest criteria. And for CPC, it's more or less the same thing, 50%. Now you can see that the picture here and here, they don't look very different. Now you have moderate here and severe here, but the picture looks very similar with the values ranging more or less between 0.1 to 10 in both cases. And this is something to note, which I will come to again later on. I forgot to mention in the earlier slide as well that those over predictions are mostly for high severity. You can see that it's either the cross, which is the ESRD, the end stage renal disease, or the squares, which is for the severe renal impairment that are outside of the twofold. The same here, whether it's AUC or Cmax, it is the severe disease and not the mild and the moderate that are mispredicted. So this is something to note. And this was exactly as here, that as you increase the disease severity, the mispredictions are also much higher. And in the mild, it's the best. The other thing we noted was that when compounds were studied clinically in more than one category, so when for some compounds we had in all three categories, for example, and then for those compounds, whenever we had good predictions in the moderate category, the hepatic impairment was also well predicted or within twofold in the other two categories. And this gives the confidence that when we only have data, clinical data for the moderate, then it's easy to use the model to extrapolate to the mild or to the severe disease population. So as before, whether it is the AUC or the Cmax, the picture looks very similar. And this is true for the liver impairment as well. And you can see that in general there are no under predictions, there are over predictions, and with increasing severity of the disease, you have more mispredictions or over predictions because you don't see under predictions at all. And one noteworthy outlier is this one, which you don't see in the other cases, only in the mild. And this is a compound that is almost entirely metabolized by the CYP3A and also has a very high clearance. And this means that there is this is possibly because of gut metabolism. You know, when you have high clearance and most of it is CYP3A, then you could expect a very high contribution from gut metabolism. And maybe the model, the way it is considering, we know that in liver impairment, the duodenal CYP3A is altered, and the way the model has captured this with the different disease severity is probably not, very good, or at least for the mild, the way the CYP3A duodenal enzyme CYP abundance is altered in the model may not be representative of reality. So now again, to focus on the outliers, compound-related factors such as extent of clearance and pathways could explain some, but not all outliers. So we had shown that the outliers were mainly coming with increased disease severity, and they were mostly over-predicted and not under-predicted. And the clearance, whether it is hepatic or renal clearance or intestinal clearance, that seemed to have some impact on the outliers. Our evaluation identified a tendency to over-predict the effects of impairment for some compounds. These over-predictions were more common in high disease severity. So this is what we have seen. And models predict linear changes in the effects of OI, or organ impairment with disease progression, whereas the observed data is not linear. So you can see that we try to plot mild, moderate, and severe whenever we had all three data, and you can see that they're not linear. They tend to kind of plateau down a little bit with increased severity. And this may be because of compensatory mechanisms with increased disease progression. On the other hand, we must also remember that the childhood classification is based on disease biomarkers, clinical features like encephalopathy, or lab-based parameters like bilirubin and so on. So this may not be necessarily reflecting the changes in metabolic capacity, and therefore either of these factors could result in the fact that there is not much of a difference in terms of metabolic capacity between the moderate and the severe. And so it's more or less similar. And this is what I had also told you when I showed you the plots, that between the moderate and severe, the picture looked very similar. Now, some perspectives on future directions from our work. We must remember that variation in outcomes could be there because of different analysts conducting the work. And the compounds in our study had very little transporter activity. Most of the compounds were enzyme driven. And this means we could not use the study to understand the impact of reduction in transporter abundance and activity for transporter substrates. And that means, the application, we cannot use SimCyP with any level of confidence, or at least our validation does not apply for compounds, whose elimination is mainly transporter driven rather than by enzymes. So some molecules show significant changes with severity and others do not. A better understanding of the sources of variability in outcome is needed to further improve predictions in the organ impaired populations. And even though predictions could be made, the reasons for outliers were not always very clear. We try to do some analysis, but we can't explain all of the variations. So known gaps in version 15 were pointed out right at the beginning, what the model captured and what did not. And I also had a conversation recently with SimCyP developers who told us that the version 15, after the version 15, there have not been many changes to the organ impaired populations, but now in the next version, they might bring about some changes. So for lipophilic compounds that depend on bile solubilization for absorption, a reduced bile acid output in the liver impaired population may lead to a reduced drug absorption. And that could explain some of the less than 1 observed AUC ratio. So this is also something, that Simsip could possibly improve in the future, or any other PBPK model could think about this, because, as I said before, the protein binding could explain some less than 1 AUC ratios, because with organ impairment, whether it is hepatic or renal, the protein binding is impacted. And that means you have more compounds available for clearance. And that means you will have an AUC ratio. That means in the impaired population, you have less exposure compared to the healthy controls with the AUC ratio less than 1. And but then, all of the less than 1 drugs, you can't explain everything just with protein binding. So for this additional, there could be some additional contributions, and we think that this could be one of the reasons, and also, as we pointed out before, accurate staging of hepatic insufficiency based on appropriate imaging and non-invasive biomarkers rather than disease biomarkers that is currently prevalent today, especially for hepatic impairment, may be needed for improved predictions. So this slide focuses on potential applications of PBPK models for organ impaired population. The recent FDA draft guidelines that came out last year for the renal impaired populations recommends an early characterization of the effect of renal impairment of a drug's PK. And this could be based on early clinical studies or on modeling and simulations, including PBPK. So this is what this draft guidelines suggests. And why do we need this early characterization? Because that will enable inclusion of the organ impaired population in late phase trials in order to broaden the eligibility criteria. So this is one potential recommendation from the FDA. Here in this picture, we can see a more comprehensive, where we think it can be applied much more. And whenever the 90% prediction interval of the impaired to healthy exposure ratios are within the bioequivalence limits range of 80 to 125%, and that is shown in this figure. So, so we, if the prediction interval, you have a AUC ratio and the prediction interval is within this bioequivalence limit of 0.8 to 1.25. So that means the effect size, the organ impairment for this particular drug is not substantial. And since our models usually over-predict the effect rather than under-predict the effect, and the effect size, even with this over-prediction risk, is small, and that means there is very good reason to believe that you can request for study waiver, especially if the drug has got a wide therapeutic index. So if you do not know what is the therapeutic index of the drug at that at any stage, and you are planning a study, then such a study can be planned later. It can be postponed because the effect size is small, even with the risk of over-prediction by the model. And therefore, you can use that to justify postponing the study to as late as possible when there is a higher probability of success for the NME. And for this study, when it has to be done, even at a later stage, we can design the study with PBPK. It can be a lean confirmatory study, and the design can be with PBPK models. And as we had seen here, you can broaden the eligibility criteria using PBPK model. You can also use PBPK models to supplement limited clinical data. And I think our analysis has given us reasons to believe that we have enough confidence to be able to supplement limited clinical data I talked to you about how, when we had data on drugs for all different disease severity classes, then when it was predicted well within twofold in one, then it was also predicted well in the other classes. And that gives us the confidence that you can supplement limited clinical data. You can also use simulations, to contribute to a totality of evidence along with mass balance, along with DDI studies and so on. So what happens when the effect size is large? It's not within the equivalence range. Then again, depending upon the indication, in oncology, for example, you can still justify study waiver when the magnitude of change is small relative to its therapeutic index. And again, you can use it to negotiate the timing of the study and to optimize the study design, broaden the eligibility criteria, and also to supplement clinical data as well as to contribute to a totality of evidence. So what are the take-home messages from our work? First of all, PBPK model predictions were within twofold of the observed data in greater than 90% of the studies in renal impaired and greater than 70% of the studies in hepatic impaired populations. And those outside the twofold error tended to be over predictions in subjects with severe impairment. Given this tendency for PBPK models to overpredict rather than underpredict, when PBPK predicted effect size is small, it is reasonable to restrict organ impairment studies to narrow therapeutic index drugs. In the absence of any information on therapeutic index of a drug, PBPK models can be used to contribute to a totality of evidence with mass balance, absolute bioavailability, and DDI studies to support inclusion of organ impaired subjects in late stage trials with or without dose adjustment. When PBPK predicted effect size is outside the bioequivalence limit, then model predictions are likely to represent a worst case scenario and may be used to design the confirmatory studies. Confirmatory studies in OI patients may be performed with a lean design, and such confirmatory studies may be considered later in development rather than early. In subpopulations where the recruitment of adequate numbers of patients are difficult, like in oncology or pediatric, our evaluation supports supplementing limited clinical data with model predictions. So that would be my last slide, I think. And with that, I am ready to take any questions now.
Thank you very much, Sheila. That was excellent. We do have some questions that have been entered into the chat boxes. Let's start here with a question from Kevin Dement. He asks, did you look at predictions of volume and distribution and how did that turn out? A very interesting point, I think.
Yeah, I don't, because we use the clinical data. I think the way it was done was, use the later time points, when we are outside of the absorption phase, and used a predicted volume of distribution. I don't think we have really done. And maybe Tycho or Steve, if you think, as far as I know, we haven't done the volume of distribution predictions, right?
Yeah, that's not something that we chose to collect. I think it falls into the category of, hindsight changes things, and perhaps something we wish that we would have focused on. But I think it was sufficiently challenging just to get the AUC and Emax data together for these compounds.
Yeah.
Yeah, but undoubtedly there are changes in volume that can take place, not just due to changes in fraction and bound, but also influences on transporter uptake. And one of the key issues in these populations is how the half-life changes and where the dosing frequency needs to be adjusted. So clearly it's an important question, and I think whoever does the next version of this can hopefully include that.
So since we don't have the IV data for many compounds, it makes it that much difficult to see whether the model predictions can be validated. Yeah.
Right. Yes, certainly. I'm not aware of IV dosing in a disease population. That's for sure. But anyway, great question. A question from Gossett Campbell: are the PBPK analysis a reflection of the stage of the disease?
I think the model predictions are, taking into account different disease stages. So the model is also adjusting the CYP abundance or protein binding depending upon the disease severity.
Yeah. So each stage of the disease has a specific tailored model in SimCip with the appropriate changes. So I think the answer is yes. The analysis did capture the different stages of the disease. Next from Jitendra Kanodia. In the renal impairment analysis, there weren't any drugs where the AUC ratio exceeded 3. Can you comment on why?
Yes, the effect size was rather low, and this could also be because the renal clearance does not play a huge role. And I think we had this one example where the metabolite is likely to have a high renal clearance, and then we did have a high large AUC ratio observed. So I think basically most drugs do not have a very huge contribution from renal clearance, and therefore the effect size is quite small.
Yeah, and I think by design, if we go back to the initiation of this, it was when the FDA were first expecting us to do effect of renal impairment on highly metabolized drugs, and so we kind of focused in on that. So the drugs we picked out, we focused more on those that were metabolized than those which had a high dependence on renal clearance, and so that's why we really don't have any big changes in renal clearance in this particular data set.
Yeah.
Yeah. Okay, a question from Sebastian Herta. In the end-stage renal insufficiency group, was dialysis used, and how is that taken into account?
In the end-stage renal disease, first of all, we had very few compounds, right? Maybe only had 3 drugs in this category.
Yeah. My suspicion, we did not specifically study the effect of dialysis for sure. We were taking studies that the sponsored had conducted, and I presume if they had done an end-stage renal disease group, they would have done it in an off-dialysis day. So even though dialysis is clearly an interesting question to address, that was really not part of this data set.
Yeah.
Right, and this is...
Yeah, I think the recent guidance requires the effect of dialysis, right? I mean, when they're on and off dialysis to be studied, but at least we only had 3, and we didn't look at that.
Tycho, you had something?
Yes, I was just going to say that it's very difficult to get patients who are end-stage renal disease who are not on dialysis, right? So that is inherently difficult to collect the data. And even from the consortium where we have so many companies, I didn't see any big data sets where we could look at the impact of dialysis.
Right. Okay. Question from Jan Stefan van der Walt. You mentioned or alluded to the shortcomings of the Child-Pugh classification. Did you think of any of the other possibilities, such as the NCI or MELD scores for classifying hepatic impairment?
No, we haven't looked at that, but maybe yeah, that is something for another time. But we haven't looked at that. Maybe other people have had some experience there. You can share, but we haven't looked at it.
Yeah, not something that we put in scope for this, but although yes, possibly something that we could come back to.
Yeah.
Yeah. And you know, to expand on that, if we go beyond then the Child-Pugh or even these other composite scores, are there or is there likely to be biomarkers that improve our ability to predict specific pathways?
Yeah. That would be difficult. I mean, I think there is a suggestion from the FDA to use sensitive CYP3A, for example, substrates. But that's for CYP3A, but to understand the disease severity rather than going by these classifications based on disease.
Mm-hmm.
But that's, I think, I don't know how many drugs there are out there that have ever been tried that with that kind of methods.
Yeah, it's a tough one because we know different pathways are affected differently by the severity of disease. And so, you know, any one marker is really going to suffer. But I guess maybe we can come up with a CYP3A4 marker and a CYP2C9 marker or something like that in the future. Yeah. Okay, a question from Manthina Varma. Any thoughts on why low clearance compounds are over-predicted in hepatic impairment while the higher ones are reasonable?
I think it is to do with the blood flow driven, right? I mean, so the low clearance ones, there is much more room for clearance to impact the exposure. If you're talking about very high clearance drug, you know, then we haven't exhausted the full capacity of the enzymes for those drugs because it's blood flow limited. And so there's not much of a leeway there because it's not entirely dependent on the clearance itself, but the blood flow. And there's a huge capacity that was not seen even in the normal patients because of the blood flow limitation. So I think it's the low clearance where you can really see the full impact of clearance and the organ impairment.
Okay, thank you. Question from Jose Mantilla. Are the changes in CYP expression modeled according to RNA data or activity data?
I'm not sure how it is done in SimCyP. I suppose it should be, you know, I think it is mostly abundance driven. You know, the changes, the only changes are abundance changes, as far as I know.
Yeah, it's certainly not RNA driven.
No.
I think it's either abundance or activity.
Yeah.
I think
I mean,
actually.
Yeah.
Because again, abundance would not necessarily tell you if activity.
No. But I don't know. The activity should actually come from some measurement in these patients. I don't think, at least in the slides that I showed, we don't capture that. I mean, it's only about CYP abundance that is, you know, easy to take care of. The activity would depend upon the compound itself, and SimCyP may not be accounting for that, I think, at least.
Okay. You know, I think, Sheila, your problem here is you finished early, so you've left time for lots of questions.
Ah, okay. Okay.
So another question from Jitendra. 90% confidence intervals with small numbers of subjects are typically not as narrow. Are the 90% confidence intervals from the model predictions narrow enough to allow this in multiple cases?
Yeah, so that 90% confidence must be within this range. And that means we're talking about compounds which do not have a huge variability in the first place, right? And also, because that has to be then within this bioequivalence range. Basically, you know, this is the minimum variability you can expect anyway in compounds. And so this is not so much. So when the idea is when the model prediction for the 90% confidence interval is within this, then the effect size is so small that you can afford to request for study waiver. I think that should be the fact that this 90% should fall within this automatically implies that the variability for such a drug in PK should be quite small. So I think it's sufficient.
Okay. A question from Xiying Chen. What is the prediction accuracy for the Cmax change in hepatic impairment subjects?
For the Cmax in hepatic impairment, is it?
You're right, right.
Yeah, yeah. I think the Cmax had very similar to the other one as well. You can see the two pictures are not very different, but it actually looks a little better than that. So for the AUC ratios, I have the numbers here, but I can come back to you, and it's also in our paper for the Cmax. For the AUC ratio, it was 70% within twofold. Perhaps for the Cmax, we had slightly better numbers. I don't recollect off my head right away.
Yeah, I think you're correct. A question from Gurab Datta. Can PBPK predictions for severe disease using clinical data from mild and moderate disease be used to substitute for real patient data in severe disease for a dedicated study waiver?
Yeah, so that is what we think, at least for the hepatic impairment, because when we see good predictions in moderate, we can be reasonably confident that it is going to predict the severe as well as the mild in a good way, because that's what we see, that when we have data for drugs in all three disease severity classes, then if they are predicted well here, then they predict well in this and the mild as well as the severe. And so that actually gives us the confidence that we can actually extrapolate having this central measurement, we can go either way. Gives us the confidence. So that's something that our analysis has shown.
Okay. I think we're coming towards the end. Maybe this will be the last question we have from Jose Mantilla. It's sort of the one I guess lots of us are asking. If you were going to do this again, what changes would you like to see, both maybe in the model in SimCip, and maybe even how would you have done the data collection and analysis differently?
Okay, so certainly, like some of the questions were asked about the volume of distribution. So that is something we could look at, but I think it would be difficult for us to do. And now in this analysis, we understand that the negative or the less than 1 exposure AUC ratio can't be completely explained by the protein binding. So some of the hypothesis that we had to support that less than 1 we would be using more compounds, and for compound selection, we would probably want to include transporter substrates as well, if there are more. And I think these are some of the things we would want to look at. Steve and Tycho, do you have other ideas?
Yes, thank you, Sheila. You can hear me, right?
Yeah.
I think this is an interesting and very relevant question, because if we ask ourselves, for example, why do we still need to run renal impairment studies? We have provided some answers here that a lot of the outcomes can be predicted, although our data set was limited to up to 45% renal clearance. So if we had to do it over again, probably we would like to include additional compounds with a higher percent of renal clearance. And then again, we would perhaps answer the question most directly if we're just running a study which is mild renal impairment or mild hepatic impairment. Can we with high confidence predict the outcome for the other extreme, Sheila, that you had talked about? And so that's one aspect of including the renal compounds with higher percent renal clearance. But the other one is, you mentioned this supplementation. I think it was on slide 14. The supplementation of limited clinical data, because as you know, we typically have maybe 6 or so subjects in dedicated studies. But as was alluded to earlier, end-stage renal disease is very difficult, or any severe hepatic impairment as well is sometimes difficult to get the patients. So we in IQ, I think, should also now look at how can we have a lot of case examples where we have less subjects and we are supplementing the maybe 2 or 3 subjects in a given cohort with PBPK modeling, so that we really have had the influence. I think there's still reluctance from companies to test out the modeling. So again, I think we should also look at if we have 1 or 2 subjects as opposed to 6, and if the outcomes would be similar.
Steve?
Yes. Well, I was going to ask if we're at the top of the hour, a little over, do we need to stop, or do we have a few more minutes?
Yeah, so I was thinking that you might have some ideas here, how you might want to do this if you had to do again.
Yeah, I think, if we look back at how we did this, where we had each company build their own models and submit them maintain the confidentiality of all of the data, that ended up being quite a constraint on our flexibility. To do, say, a volume distribution question, it was going to be quite a lot of work. So I think a different way of doing it really would have given us a lot more flexibility.
Yeah.
I think one of the features of SimSip in version 15 was that the shunting in hepatic impairment is only available for the full PBPK modeling, and not all of the compounds were modeled using that approach. I know SimSip will be adding that to the partial PBPK modeling part of the software in the next version. So, that sort of difficulty will be addressed, and I think that will be very, very helpful in making these analyses a bit more tenable going forward.
Yeah, I think the validation, I have a feeling different analysts could have contributed to, some kind of bias in the prediction as well. So across validation, if somebody's doing the work, somebody else to make sure that it's exactly coming in the same way would also be helpful, because the user bias cannot be completely eliminated in this approach, as I pointed out.
Yeah, great. Okay, well, I think we're at the end, and it's time to wrap up. I think this has been an excellent discussion, and hopefully if you have questions that you would like Sheila or any of the others to answer, you can do so. I think you have their email address on the slides, and we can take care of it that way.
Thank you, Mahalia, and thank you, Steve and Tycho.
Yes, okay, great. Take care. Bye.
Thank you.
Take care. Bye.